I sometimes ask my classes at some point to name an animal. I just start, oh, everybody, let's name an animal and we'll stop when we get to 10. And we just go around the room, people start naming animals, and typically I'll hear cat, dog, person, giraffe, etc. And in some classes, all of the top 10 that leap to people's minds are mammals. Sometimes someone will say bird or snake or something, in which case everyone that had been named was a vertebrate, and actually a vertebrate which lives on land. Uh, if you were to consider, what animals actually are, however, you realize that we have a bias. So when you think of the word animal, if you're thinking of dog or person or bird or bear or something like that, you have a bias. Because, for example, if you were to take just the sheer mass of animals on Earth, um, wild birds, it is estimated, make up, say, 2 megatons. Wild mammals, 7 megatons. But the nematode worms, the type of worms that most people don't even know exist, many of them are microscopic, and we walk on them when we, step in soil. They outweigh both of those by multiple times. There's actually a lot of people on Earth, and a lot of livestock that would include cows, pigs, chickens, so birds and mammals here. So that's a whole other topic. But when we talk about cnidarians, this would be corals, jellyfish, etc., which we'll get to in this lecture. There's 100 megatons of them, equal to the weight of all of the livestock. When we get to segmented worms and mollusks, there are more. There are certainly a lot of fish. Those are the most abundant vertebrates. Notice the number of fish compared to the number of mammals, but then notice the number of arthropods. Arthropods being the major group of life on Earth. And if you were to exclude fish, equaling the mass of all other groups combined. Now, arthropods are obviously small, so when it comes to individuals, not only do invertebrates just weigh a lot on planet Earth, but in terms of individuals, they make up more than 99% of the animal individuals and well over 95% of the animal species. So point is, if as biologists we're going to consider animals, we have to start with invertebrates. They are the most common animals, whether by numbers of species, or by weight. As we'll see, they were the first animals. So a lot of the features of animals evolved first in invertebrates. They make up the basis of food chains. So certainly, that is where we need to start. Now, just a tiny review of the previous topic of protists. So if you were to take all of the groups of life on Earth and say, all right, when do they exist in the fossil record? If I start finding old rocks, where do animals first appear? Where does life first appear? We realize that for a while there is no life on Earth. Our planet is a dead planet. And then there is life starting 3.5 billion years ago, maybe a little earlier. But for about a third of the history of life, the only life on Earth are bacteria, the prokaryotic cells. And then the blue-green algae start adding oxygen to the atmosphere. And shortly after, we get a new type of cell, a more complex eukaryotic cell, which we then call many of them protists. Animals don't appear until much later, as we'll see, maybe 700 million years ago. Now, I'd like to talk about protists for just a second, because protists include all of these diverse groups. So amoeba and brown algae and red algae and euglenozoans and dinoflagellates and diatoms, etc. And then there is a group called the choanoflagellates. Most people don't know about these. These are a very minor group, but they have a very interesting shape, because in a second, I will be discussing the choanocytes in sponges, which look an awful lot like choanoflagellates. And so whether it be in morphology or in genetics, one of the very important new genes which make animals a group and let them diversifies a type of protein called a tyrosine receptor kinase, or I'm sorry, receptor tyrosine kinase. Animals have them, but so do choanoflagellates. And so sponges have their most abundant cells are very similar to the choanoflagellates, the type of cell that composes these choanocytes, which have flagella. And so there were protists which had some animal features, and they exist today. And we could then get into interesting discussions because these choanoflagellates, they are not multicellular, but they do sometimes hang out in groups, form small colonies. And therefore, one of the interesting questions that we could ask is this type of colonial organization the precursor to multicellularity? So in any case, choanoflagellates are a group of protists which are more closely related to animals than any other protists are. And then once again, the word protist, as a biologist, I have to cringe a little when I use the word because we like to use names of groups which signify this is a monophyletic group, a clade. So for example, all eukaryotes are descended from the very first eukaryotes. So eukaryotes is a good group. But if you were to ask what groups do we see here, well, there's certainly, lots of, separate lineages we could talk about. But, if you wanted, names to introduce into, say, general biology classes for people who aren't specializing in protist classification, the difficulty here is if you were to ask, is this a real biological group, these protists? And the answer is no, because some of the descendants of the first eukaryotes are called plants, and they are most closely related to the green algae. The green algae are more closely related to plants than they are to other groups of protists. The fungi are more closely related to some protists, like microsporidians, than they are then the microsporidians are related to other protists. And the same way, the choanoflagellates are more closely related to animals than they are to these other protists. So the group protist or protista, it's a paraphyletic group. It includes some, but not all of the descendants of the common ancestor. It doesn't form a good group because if it was a complete group, then it would include animals, fungi, and land plants. And so then talking about protists as if they are a group is not representing biological relationships, since these choanoflagellates are more closely related to animals. So with that, then we get to animals. And it makes sense to start with the simplest animals. It also makes sense to start with the first animals. And as one might expect, you can do both of those at the same time because the simplest animals were the first animals. So if we look at the history of Earth, we separate it into eons, the Hadean, the Archean, the Proterozoic, and the Phanerozoic. Virtually all animals are confined to the Phanerozoic eon. So all vertebrates start in this purple region, which begins 540 million years ago. All arthropods are known from this time period, all mollusks, etc. But just before this Phanerozoic eon began, there was some cases of animals being known. These would be the simplest animals: sponges, cnidarians, and worms. And so if we were to then go to the geologic history, we could start with the simplest animals then also being the very first animals. And so by what was known the Ediacaran period, and maybe slightly before, the Ediacaran period starting maybe 630 million years ago, this is when the cnidarians are the most common animals on Earth, the most diverse animals on Earth, and there are also worms, etc. And then it would be in the Phanerozoic eon here in purple that we then split into eras, the Paleozoic, Mesozoic, and Cenozoic. This is when we would get arthropods and fish known from the Cambrian, etc. Okay. So what is an animal? Well, there are definitions. And so when we use, once again, biological terms, animals are a group, a real biological group. It's not some random assortment. And the reason it's a biological group is that all animals share features which the non-animals lack. For a while, it was thought that those receptor tyrosine kinase enzymes, very important in animals, were one of those features. But then it was found that the choanoflagellates have them as well. Another feature which was thought to be unique to animals was collagen. Here, that's what is going to be found outside these cells. And while some of the uses of collagen and its abundance are unique to animals, actually a little collagen is known in fungi. And if you look at that Protistan family tree, you might have noticed how close the fungal and animal branches were. So the earliest animals, they achieved multicellularity. The individual is the group of cells, not the individual cell. And so if you look at, say, an amoeba or a choanoflagellate or a paramecium, the individual cell is the individual organism. But if you look at a plant or most fungi, or if you look at an animal, the individual is not the cell, it's the group of cells. We are multicellular. Fungi actually do have some unicellular members, the yeast. So the first animals then learned to depend on each other so that it was the group of cells which formed the individual. One of the things that helps them to form that group is collagen. All cells have proteins on the inside of them, but what makes animals special in this case is they make collagen as an extracellular protein. And so not only are there proteins inside the cells, but now there are proteins outside the cells. And this can form a matrix to which the cells adhere. And so then a lot of the genes which would then get established in animals would be genes for linkages between cells or genes to interact between cells. So cell membrane receptors, proteins which allowed for junctions between cells, etc. So if you look at the sponge here, the sponges are the simplest animals known. They're called the simplest animals known because they don't have tissues. They are multicellular, all right? So they have that level of organization where they're multicellular, but they lack tissues, they lack organs and systems. All other animals other than sponges have tissues. And so notice that sponges are multicellular, and in the human body, the most abundant protein is collagen. In the body of a sponge, the most abundant protein is collagen. And that's true of all animals. So collagen makes up more of an animal's weight than any other protein, and that includes sponges are the simplest animals. Now, while plants are doing photosynthesis to obtain their energy, and fungi are often decomposing dead material, animals, they cannot do photosynthesis unless they have endosymbiotic algae living inside them, like the corals, which we'll get to. And so animals have to ingest other living things. All animals then are predators to some degree. Now, some we would call, parasites, but they're not getting energy from the sun, or by oxidizing minerals, or by decomposing dead things. So their energy has to come from the molecules of other living things, and their carbon cannot be fixed from carbon dioxide in the air. This carbon that I use for my sugars and proteins and lipids, etc., this was then came from the carbon that I ate. All right, so when I ate salads or cheeseburgers or macaroni and cheese or banana bread or whatever, that was biomolecules, sugars, proteins, etc., that came from other living things. Lettuce, bananas, livestock, etc. And I now have incorporated those molecules into me. That's where I not only got my energy to get through the day, but also the biomolecules that make me up. So animals, whether it be this great blue heron eating a catfish, or this microscopic rotifer, which is filter feeding bacteria and protists out of the water, these animals share that trait. They are multicellular. Collagen is their major extracellular protein, and they have to ingest other living things. That's how they get by. Okay. So animals are not known for most of the history of life on Earth, and it's really around 700 million years ago that we get the first traces of very simple animals, but most of the animals then appear much later, as I'll be discussing throughout these lectures on invertebrates. Once again, sponges are the simplest animals, and although we could argue over some of the fossils, apparently it would seem also then the first animals, or at least among the first animals. So here we can see some modern sponges, and there are fossil records which have been classified as sponges going back about 700 million years, which is earlier than any other animals. So sponges, the simplest animals, and then also it would appear the first animals. Here you see sponges. They are filter feeders where water is coming in, water leaves, but in the process, they are trying to trap bacteria, blue-green algae, obviously, which are cyanobacteria, other forms of algae, protists, small animals, etc. And so that's how they make their living. So they are ingesting other organisms in order to survive, as do all animals. Now, I just have a little aside here because we could argue over the first, the earliest fossils of these. Now, it is frustrating, and certainly as a biologist and a teacher of biology, it is frustrating because you'd always like to say, this is the facts, this is what the evidence supports, next. But that's not how science works, right? In science, science is an attempt to understand the natural world, and you make hypotheses, you test them, and here's the evidence. But then someone can make another hypothesis, and there could be future evidence which kind of undermines previous evidence. So when are the sponges first known in the fossil record? Well, if you search the scientific literature, you can find records of, here are sponges going back 700 million years. Here are sponges going back to the Ediacaran period. And so if you were to look at the late Proterozoic, you could speak of the Tonian period, where perhaps there are fossils of sponges. There's that period of snowball Earth when there were really severe ice ages in the Cryogenian period. And then in the Ediacaran period, where now we start to get things like jellyfish relatives and worms. In all three of these, there are fossils which have been identified as sponges. However, some individuals might reinterpret this and say, I'm not sure I would call that a sponge. I mean, we are dealing with the fossil impressions of invertebrates, which are largely lacking in hard parts and are often very small. And they say, you know, I interpret this fossil a little differently. And so while there are sponges which we can definitely classify as sponges and related to either an extinct group of sponges known as the archaeocyathans or the groups of modern sponges alive today. These are certainly known from the Cambrian period. Somewhere in here is the Cambrian, there we go. And so by the Cambrian period, they are certainly present. Sponges seem to be, there have been reports of sponges in the 3 periods prior to that, in the Ediacaran, the Cryogenian, and the Tonian. And once again, these are published scientific journals, and so once again, I would refer you to those. But then some individuals say, you know, I question this fossil, etc. So there is evidence to suggest that sponges were the very first animals, and when you do genetic comparisons, sponges then branch out as the earliest or one of the earliest groups of animals, being more primitive than cnidarians. And so once again, whether it be genetic studies, fossil studies, anatomical studies, you know, the consensus would be that sponges are the simplest animals and therefore candidates for the very first animals. Okay. And so if you were wondering, you know, what animal life first looked like, there would have been a time on Earth, arguably then, that there were no jellyfish, there were no worms, there were certainly no fish or arthropods, and sponges were the only animals on Earth. Once again, how do sponges work? Water comes in through these openings, which are known as ostia, and then comes out the larger space here, the osculum. Right, so water, you know, comes in, water leaves, but then while it is there, that these choanocytes, very much like those choanoflagellates, as I said, they are creating a current by their flagella beating, and then this allows things like cyanobacteria, green algae, etc., to become trapped. Right, so most of the cells here, so that one, 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 these are all choanocytes whose flagella are then creating those currents. Now, sponges are ingesting cells through phagocytosis. So this cell has to perform phagocytosis of any ingested food. And as a result, then, food size is, you know, limited to what can be ingested inside a cell. I mean, other organisms will have a mouth or a gastrovascular cavity which can hold something larger. So, you know, jellyfish can feed on fish, for example. But here, because these cells can only ingest these smaller things through phagocytosis, as sponges are then limited. So sponges are multicellular, and the majority of those cells are those choanocytes, which are creating the current, which is how they feed. Now, we'll notice a few other things. There are spicules, which we see here. These can give the sponges a more solid structure. So in addition to collagen, they can have inorganic spicules, and then there can be different kinds of sponges. So if silica is used, for example, in glass sponges, then these can have different chemical formulas in different groups. These cells look like amoeba, and they can go around and say ingest bacteria, which among other things protects sponges from infection. And that's interesting because my white blood cells can move like amoeba and protect me from infection. In fact, all animals possess amoeboid cells, which then protect from, among other things, they can ingest microbes, but this has an added feature of protecting from infection. So this seems to be an early feature of animals. Sponges can reproduce asexually, and so this can occur through fragmentation. And so if a sponge simply breaks into pieces, now these two pieces can then be now isolated. But sponges can also produce sexually, so they can produce sperm and ova, which then can be released into the ocean water, and so fertilization can then occur, and can actually occur, outside the body of the adult. Now, when we look at sponges under a microscope, okay, some of the cells that we're looking at here, these larger cells, those are ova. Here is a microscopic view of sponges, and these large cells, these are the cells which have undergone meiosis and are producing ova. So a couple of things. One, in addition to reproducing asexually, sponges can reproduce sexually, as you can see here in the production of these ova. But then also notice how primitive sponges are, because all other organisms have what we call gonads. There is a specific region of the body where meiosis occurs and sex cells are produced. Whereas in sponges, this can occur anywhere in the body. There isn't a specific region that sperm and ova are only produced in these regions. So sponges can also reproduce sexually, but once again, they are the only animals which lack gonads. One final point is that as we look at sponges, the groups we have today have evolved over time. And so those, Precambrian sponges are not in modern groups. When you get to the Cambrian, there's all of these primitive sponges. But then there is a group of sponges known as the archaeocyathans. They are only known from the Cambrian period. So this is a group of fossil sponges, which would have been quite important on Earth because at the time of the first sponges, there were no coral reefs yet. And so sponges would have been the major reef-building organisms of the early Cambrian. And so thus they would have been very important for marine life. These archaeocyathans, however, they became extinct by the middle of the Cambrian. And so thus their appearance and then their loss probably would have greatly affected other marine animal life. So while that group of sponges became extinct, then obviously some have survived till the modern day. And here you can see modern sponges which are diverse. There are diverse groups. Notice the different spicules making up these sponges. And sponges continue to form important parts of ecosystems today. So they are filter feeders, but they also perform some photosynthesis. Some of them do have endosymbiotic algae, and they are a reef-building organism. And so while things like corals are the major reef organisms of today, sponges play a role as well.